Hello students, welcome to EPG Pathshala. I am Satish Manchanda from DIPSAR, that is Delhi Institute of Pharmaceutical Sciences and Research, New Delhi. Today we are going to talk on the module that is Characterization of Microspheres from the paper Novel Drug Delivery System 2. Let's see what are the learning objectives of this module. First one, we are going to study about the evaluation of microspheres. Under this, we will study physical characterization of microsphere, chemical characterization of microsphere, thermal characterization of microspheres, and biological characterization of microsphere. After that, we are going to learn about the applications of microspheres. Let's study about the characterization of microsphere. Microspheres have different microstructures which determine the release and stability of the carrier. Thus, the characterization of microspheres is important in order to design a suitable carrier for drug delivery. The characterization of the microspheres can be done by checking the different parameters that are chemical, thermal, and biological parameters. Let's start with physical characterization. Physical characterization of physical parameters. Physical characterizations are classified into several parts, that is particle size and size distribution. Next is surface morphology. Next is surface charge, and then electric surface potential and surface pH. After that, density and drug loading. After that, we are going to study about drug release from the microsphere. Now, first of all, particle size and size distribution. Particle size and size distribution parameter is important to study because it affects the other parameters like stability of the microsphere, drug release from the microsphere, and the drug loading. The various techniques used to characterize the particle size and size distributions are: number 1, electron microscopy, then size exclusion chromatography, optical microscopy, and sieve analysis. Now, first of all, electron microscopy. The electron microscopy techniques include SAM and TAM. SAM is expanded as scanning electron microscopy and TAM is expanded as transmission electron microscopy. This technique uses the beam of electrons which interact with the sample, thus producing signals which is detected and provides the sample information. SAM is based on the scattered electrons, whereas TAM is based on the transmitted electrons. SAM provides the information about the topography of the sample surface and its composition, whereas TAM provides the information about the internal structure of the particles with high resolution power than the SAM. Size exclusion chromatography. This technique works on the basis of molecular size of components to be separated. In this technique, samples are passed through the bed of porous particles and hence by the differential exclusion form from the pore separation occurs. Size exclusion chromatography is a widely used polymer characterization method because of its ability to provide good molar mass distribution results for polymers. Physical characterization under this, optical microscopy. This technique is based on the simple ordinary microscope technique. Here, an emulsion or suspension, diluted or undiluted, is mounted on the slide or ruled cell and placed on mechanical stage. The microscope eyepiece is then fitted with a micrometer by which the size of the particle can be estimated. Next technique is sieve analysis. Sieve analysis is performed using a nest or stack of sieves where each lower sieve has a smaller aperture size than that of the sieve above it, which is allowed to shake for the particular time period. Thus, the mesh size of the sieve through which the particle passes and through which it does not pass give the rough idea of the particle size. Sieves can be referred to either by their aperture size or mesh size or by their sieve number. Physical characterization under this surface morphology. The various techniques used to characterize the surface morphology are SAM and TAM. SAM stands for scanning electron microscopy and TAM stands for transmission electron microscopy. These two techniques we have already discussed in the previous modules and previous sections. Another technique used are atomic force microscopy, that is also termed as AFM, and confocal laser scanning microscopy. Physical characterization. Let's start with atomic force microscopy. Atomic force microscopy, that is AFM, is a powerful tool for surface and morphological analysis. AFM uses a sharp tape to probe the surface features by scanning and can image the surface topography with the extremely high magnification up to 1 million x, comparable or even better than electronic microscopes. Next technique is confocal laser scanning microscopy. Confocal works similarly to that of SAM, that is scanning electron microscopy. However, it is able to provide the internal structure information provided that the particles are transparent and can be fluorescently labeled. Physical characterization. Next parameter is surface charge. Surface charge plays an important role in the stability of the microspheres as well as in the circulation time in the blood, thus affecting the bioavailability of the drug. Negatively charged particles are more subjected to opsonization process by RES system of the body. However, for better stability in the colloidal system, charged particles are more stable as they have the tendency to repel and hence no agglomeration occurs. Surface charge can be measured by free flow electrophoresis or by measuring zeta potential. Physical characterization. Next parameter is density. For the determination of density, different techniques can be used. Densities can be mainly of different types. For example, true density and granule density. Now, true density. It can be determined by helium densiometer or can use liquid displacement technique with the help of pycnometer. However, helium is able to penetrate the smaller pore and surfaces, so helium method gives the closest approximation to true density. Next is granule density. It uses the technique similar to liquid displacement technique. However, mercury is used to fill the void spaces and fails to penetrate the internal pores of the particles. Physical characterization. Another parameter is flow properties. Flow properties play important role in designing the tablet, capsule and other dosage forms. Similarly, when microspheres are used to prepare tablet or capsule dosage form, flow properties need to be measured. Angle of repose is one of the parameters which can be measured to check flowability of microspheres. Rougher and more irregular the surface of microsphere, more is the angle of repose with poor flow. the other parameter is drug loading. The drug can be loaded into microsphere at the time of preparation of microsphere or after the preparation of placebo microsphere. Drug loading can be checked either by dissolving the formed microsphere in suitable solvent and then calculating the amount of drug present, or by checking the amount of drug left in supernatant after centrifugation, especially for incubation method of drug loading. Drug loading can be calculated as the amount of drug in microsphere divided by amount of microsphere multiplied by 100. The unit may be milligram or microgram. Entrapment efficiency. The entrapment efficiency can be determined by measuring the amount of drug that is encapsulated in microsphere. The percentage entrapment efficiency can be calculated by using the formula, which is amount of drug in microsphere in milligram divided by initial amount of drug in milligram multiplied by 100. The result will occur in percentage. Drug release. Drug release can be determined either by the use of dissolution apparatus or by the use of dialysis bag. Dissolution apparatus basically uses paddle and is carried in similar ways as for tablet or capsule dosage form. For dialysis, first the activation of dialysis bag is necessary, which is done by treating with different chemical reagents, and then the drug-loaded microsphere suspension is placed within the dialysis bag, and the bag is placed in the receptor compartment, which is under stirring and set at particular temperature. Sink condition is maintained and aliquots are withdrawn at different time intervals and are analyzed quantitatively with suitable analytical method, that is by UV or HPLC. The amount of drug released at different time interval is then plotted to fit different release models like zero order release model, first order release model, Higuchi model, Hickson-Kroll model, Korsmer-Peppers model, and several other models known. Now let's study about the chemical characterization. Chemical characterization of microsphere are classified into various parts, that is surface carboxylic acid and amino acid residue and compatibility study. Let's start with surface carboxylic acid residue and surface amino residue. Surface carboxylic acid residue is measured by using radioactive glycine. The radioactive is conjugated with the microsphere by using a linker, and the radioactivity of the conjugate is measured by using liquid scintillation counter, from which carboxylic acid residue is correlated. Similarly, for surface amino acid residue is determined by radioactive C14 acetic acid. This is indirect method of determination. Initially, carboxylic acid residue is measured through scintillation counter, and then amino acid residue is linked indirectly. Now come to compatibility studies. It can be checked by many techniques. Most commonly used techniques in the research are by FTIR study and DSC study. Comparing the FTIR spectrum and DSC curve of the drug, microsphere, carrier, and microsphere loaded with drug, one can identify changes in the drug molecule due to interaction with carrier molecule. Thermal properties analysis. Thermal property analysis is especially important to check for the drug-loaded microsphere. Because of the technique used in the preparation of microsphere, sometimes there is a change in the internal properties of drugs like change in crystal behavior or crystallinity. Drugs may change from a crystalline to the amorphous form, thus enhancing bioavailability. Thus, these parameters can be checked from differential scanning calorimetry or X-ray diffraction, which is also termed as XRD. For the amorphous, DSC shows no peak, whereas for crystalline structure, there is a sharp peak due to the phase transition with increase in temperature. Biological characterization. Since the microspheres intended for parental administration should be sterile, pyrogen-free, and should not produce any kinds of toxicity in human beings. Thus, produced microspheres should be tested for sterility by incubating in suitable culture media for aerobic and anaerobic bacteria and checking for the growth of the microorganism. Whereas for the pyrogen testing, LAL test is very commonly used. Application of microspheres. Let's start with the introduction. Microspheres encapsulate or entrap the drug molecules into the polymer, thus enabling controlled and sustained release. Protection of drug molecules or protein and peptide from degradation, targeting drug to the site of action, especially for the anti-cancer drug delivery by coating with suitable ligands antibody to the surface of microsphere. Nowadays, there have been developments of bioadhesive microspheres for nasal delivery system, mucosal drug delivery system, vaginal drug delivery system, especially for the drug that undergo hepatic metabolism. Poorly stable drug in GI tract, thus enhancing bioavailability of the drug. Now let's start with the microspheres for controlled and sustained drug delivery system. Polymeric microspheres are the ideal vehicles for many controlled drug delivery applications due to their ability to encapsulate a variety of drugs, biocompatibility, high bioavailability, and sustained drug release. These types of microspheres have a huge potential in drug delivery system. They enable to provide targeted drug delivery system through localized action of bioadhesion, that is, local targeting. Along with it also provides the controlled drug release of drug, which is ultimately governed by the degradation profile of the polymer, dissolution and diffusion of drug molecule from the polymer. Now next is microspheres for targeted drug delivery. Targeting techniques of microsphere is basically based on two ways, that is active targeting and passive targeting. Active targeting, in this the surface of the microsphere is linked to the specific molecular group that interacts to the specific receptor in the target tissue, while passive targeting of the microsphere is based on the physical and the chemical properties of the microsphere, especially size, charge and surface characteristic, that is hydrophobicity, so that the particles reach the desired site of action. Microspheres have been employed extensively with cytotoxic drugs, the chief example being doxorubicin, mitomycin C, cisplatin and 5-fluorouracil. Magnetic microspheres, radio microspheres has revolutionized the field of anti-cancer drug delivery. Controlled release vaccines. In recent years, research on microsphere-based vaccine delivery vehicles for single administration has got much attention. Microspheres are thought as the potential alternative in the development of single-shot vaccine which can provide the necessary booster dose at the specified time after administration, thus eliminating the need for the patient to return for boosters and would reduce the risk associated with the reuse of disposable needles and syringes. Also, the ability to more precisely control the time course of vaccine deliver may lead to more effective vaccination with current antigens and may allow utilization of antigens that were previously ineffective. Microsphere targeting for cancer therapy. Microspheres have been employed extensively with cytotoxic drugs, the chief example being doxorubicin, mitomycin C, cisplatin, and 5-fluorouracil. Cancer therapy demands specificity as to avoid damage to the healthy cells, thus targeting with microspheres is being exploited recently to achieve the demand. Targeting to the cancer cells can be achieved as discussed earlier or by the utilization of changes in the tumor environment like change in pH, accumulation of particular factors or receptors at the site of tumor. Magnetic microspheres, radiomicrospheres, has revolutionized the field of anti-cancer drug delivery. Ultra-fine particles of magnetite, that is Fe3O4, have been incorporated into both albumin and cellulose microsphere containing the anti-cancer drug doxorubicin and didox. The movement of particles can be controlled after intra-arterial administration by an externally applied magnetic field. Up to 70% of the administered dose of doxorubicin can be directed to the target site by this mechanism, which has shown success in inhibiting the growth of subcutaneous tumor. A recent patent from Japan described new technology in the form of porous silica microspheres coated with ferromagnetic ferrite. The inventors claim uses of the technology for ultrasonic diagnosis and dermatology. In particular, the treatment of solid tumors utilizing heat generated through converged ultrasonic waves. Therapeutic efficacy was demonstrated after direct injection of microspheres into the liver of rats inoculated with tumor cells. Tumors were irradiated with ultrasonic waves of 2 MHz frequency and 10 watt for 3 minutes and temperature was raised by 5 degrees Celsius resulting in tumor cell death. Diagnostic purposes of microsphere. Microspheres are also useful for the targeting purpose in which various cells, tissues and organ cell lines can be imaged by radio-labeled microspheres. Particle size analysis of microsphere is a main factor in determining the imaging of particular sites. For example, the particles injected intravenously will become entrapped in the capillary bed of the lungs. This is used in imaging of the tumor masses in lungs using labeled human serum albumin microspheres. The adequate expression of cell surface receptors and antigens is an important requirement for the functional ability of living cells. A lot of different methods for cell surface marker tracing have been described. However, most of these techniques have disadvantages, limiting their wide-scale utilization in routine laboratory and clinical practice. The most recent technique for these purposes, approaching near the ideal one, is based on the use of synthetic microsphere particles made of polymers which are formed mainly by emulsion or radiation polymerization of a variety of monomers. The resulting spherical particles bear hydroxyl, carboxyl, amino, or other functional groups capable of covalent binding of proteins, dyes, or chemotherapeutic agents. Fluorescent, radioactive, or haptanic labels may be introduced already during the polymerization too. There are three main fields of application of such specific labeled microspheres in cell biology. Number 1. Detection of cell surface markers. 2. Studies of phagocytosis mediated by cell surface markers. And number 3. The cell separation according to cell surface markers. Microspheres with different functional groups like hydroxyl, carbonyl, and aldehyde can be obtained by the polymerization techniques. These functionally activated microspheres can be used for adsorption and covalent immobilization of human and animal protein human serum albumin, that is HSA, gamma globulin, human fibrinogen, and enzymes, for example glucose oxidase and urease. Bioavailability enhancement. The above applications like targeting, bioadhesive, sustained release property is directly or indirectly linked with the enhancement of the bioavailability of the drug. Targeting enhances the availability to the site of action. Bioadhesive like adhesive eliminates the harsh condition of GIT and hence enhances bioavailability. Protection of drug by the encapsulation of drug also ultimately enhances the bioavailability. Floating microspheres enhances the residence time of the formulation onto the stomach, sustaining the drug release and thus improving the bioavailability. Also, X for targeted disease related to stomach and colon. It was reported by Jong-Soo Wu et al. For the enhanced bioavailability of poorly soluble drug cyclosporine. By formulating it in combination with hyaluronic acid into microsphere. Oral bioavailability was enhanced because of enhanced dissolution rate and bioadhesive property of hyaluronic acid. Polymer microspheres can also be used to delivery of proteins. This is a very important class of drug delivery as the release of protein from such formulations can be well controlled, thus protecting the encapsulated proteins from enzymatic degradation. A large body of literature exists which shows that small, generally less than 2 mm, thermoplastic microspheres delivered orally are able to cross the intestinal epithelium either through Peyer's patch and or the regular absorptive epithelium and travel systemically to other sites including the liver, spleen and other tissues. These observations have led to the proposition of three major mechanisms of action for microspheres for improving oral delivery of protein. Number 1, the spheres can protect proteins from proteolysis. Second, the spheres cross over the intestinal mucosa. And third is the microsphere changes the intertisular distribution of proteins throughout the organism. The last proposed mechanism can be extremely important for drugs like insulin, as microspheres are expected to build up in the liver, where loaded insulin can be used to limit the abnormally elevated hepatic glucose alone, without the use of encapsulated formulations production present in diabetic patients. Different types of microspheres with research on their application in drug delivery system. There are different type of microspheres which are having different application. Let's start with the bioadhesive microspheres. They are used in buccal, oral, ocular, nasal, and colonic drug delivery. The examples of nasal drug delivery include the gentamicin and insulin, whereas the example of GI drug delivery include glipizide. The examples of colonic drug delivery is insulin, and whereas the example of ocular drug delivery is methylprednisolone. Now let's come to the magnetic microsphere. They are used in DNA analysis, cell isolation, protein purification, and targeting the drugs to tumor sites. For example, doxorubicin. Next is floating microsphere. They are used as carriers for drugs like antiviral, antifungal, and antibiotic agents. So they are called as absorption windows. They are also used for non-steroidal anti-inflammatory drugs like prednisolone, and they are also used for lansoprazole. Let's start with radioactive microsphere. They are used for diagnostic purpose, diagnostic radioimmobilization, 99M technetium macroaggregated human serum albumin, that is termed as MAA, thrombus imaging in deep vein thrombosis. They are also used for therapeutic purpose, radioimmobilization of liver and spleen tumor. Next, 90Y microspheres, local radiotherapy, that is 212 lead sulfur colloid. Another type of microspheres are polymeric microspheres. They are used for vaccine delivery like hepatitis, influenza, pertussis, diphtheria toxoid, oral drug delivery of easily degraded drugs, also used for gene therapy with DNA plasmid, delivery of insulin, LHRH controlled drug delivery after local application, like release of proteins, hormones, and peptides over extended times. Available marketed products of microsphere. There are several drugs which are available as microsphere formulation. The examples include levodopa floating sear capsules by the brand name Medopar, which is sponsored by Roche Product USA. Diazepam floating capsules are also available by the name of Well Release, which is prepared by Hoffman La Roche of USA. Liquid Gaviscon is a brand name for aluminum hydroxide magnesium carbonate effervescent floating liquid alginate, which are prepared by GlaxoSmithKline India. Topaclane is a brand name for aluminum magnesium antacid floating liquid alginate preparation, which has been prepared by Pierre Fabre Drug from France. Ferrous sulfate colloidal gel forming FDDS gas generating floating tablets are available by the brand name of Conviron, which has been prepared and marketed by Ranbaxy India. Cifran OD is a brand name for ciprofloxacin colloidal gel forming fast dissolving drug delivery system, that is FDDS, which has been prepared and marketed by Ranbaxy India. Cytotec is a brand name for misoprostol bilayer floating capsule from Pharmacia USA. Ofloxacin gas releasing floating tablet are available by the brand name of Oflin OD, which has been prepared by Ranbaxy India. So students, let's now summarize what we have learned in this module. First of all, we can say the characterization help to optimize the particle size and morphology of the microsphere so that we can upload a maximum of drug to show its therapeutic activity. Characterization is also helpful for determining rate of drug release and stability of the microspheres. It is also helpful to design a suitable carrier for the drug delivery depending upon the desired site of action and the targeted drug delivery or organ. The application of microspheres are wide in therapeutic delivery. Like we have studied, they are applicable in ocular, parenteral, anti-cancer, etc. Also, they are helpful in targeting and controlled release application of drug in various diseases. I hope this module will help you for your studies. Thank you.